you'd like to receive emails, if you'd like to receive emails about future events like today or events like DNA Day, we encourage you to sign up on the following links from Illumina Cares team and the Biocom California Institute. Michaela from my team will be posting that in the chat box here shortly. The webinar will have two parts today, with our keynote speaker presenting for the first 30 minutes with a follow-up Q&A. And then the second half will feature resources available for teachers to use in their classroom, focused on agriculture and genomics education. Without further ado, I am honored to introduce our keynote speaker today, Khalil Lawless, who is the Global Agrigenomics Segment Manager for Illumina. From New Zealand, Khalil studied biomedical science and genetics at Victoria University of Wellington and moved on to become a molecular biologist and research scientist with the Victorian Department of Primary Industries and the Environment, working in plant genomics and genetic improvement. Now with Illumina for 10 years, spanning 3 continents, Khalil leads the marketing of Agrigenomics segment, bringing state-of-the-art genomics technologies to agriculture and conservation to help ensure food security and productive sustainability for the future. Ladies and gentlemen, please welcome Khalil Lawless. And I'm going to stop sharing my screen, Khalil, so you can share yours.
Thank you very much, Howard. Happy to be here. Thanks for the opportunity to speak to everybody today. I really hope everybody finds it an informative webinar and we can all learn. So here comes the slides. Tell me if you can see them.
You're good to go.
Okay, lovely. We'll make a start then. My name's Khalil Lawless. I'm an employee of Illumina. I'm a molecular biologist and geneticist by trade. And I'm here to help, explore some areas around agricultural biotechnology and sustainability and learn a little bit more about some of those areas, how they work and the impacts that they have, and look at these different technologies and how they can be appropriately used to tackle some of the big challenges that we have in front of us. So in order to do that, I'm really hoping that at the end of today's class, you will understand a little bit more about the global environment and food security outlook and how we've got to where we are. I'm hoping you guys can understand and learn a little bit more about how genomics and genomic technologies, not just gene editing, can be used to address some of these challenges. And then also just to explore the interconnectedness, of agriculture, sustainability, genomics, human health, right? Because all of these things are interconnected and we have to take a holistic view in order to solve these problems. And then I can hopefully, show a little bit more about the work that I do and a little more about me. And we're going to start there. Like Howard said, I'm from New Zealand and I grew up in New Zealand, which is a very beautiful place with a wonderful environment and natural spaces. And I grew up in a conservation family. And my father worked in conservation. My brothers worked in conservation. In fact, this is me here with the big fuzzy hair. Working with my brother who was researching a mountain parrot at the time as an ornithologist. And I used to help him with his field work. And it took me to a lot of beautiful and out of the way places. And then, I went on to develop my own love and interest in biology. And so I went on to study biology and genetics. I was very fascinated by evolution and how that worked. And the skills that I picked up there enabled me to go into a career in research. And so I began doing research with the Department of Primary Industries in Australia. And I was involved in a lot of crop genetics projects where we were looking at how to make certain crops and grasses that were used by livestock more resilient to environmental stresses, more productive. And then I took some time out of that also because I'm very passionate about the ocean and I spent a long time traveling. I went to South America and did some volunteering projects with wildlife refuges. I spent a couple of years sailing around the world on yachts and got to see a lot of amazing natural environment and learn a lot about the state of our oceans as well. And then I came back and restarted my career in science and was actually involved in some of the very first gene editing experiments that were ever conducted in Australia. And based on that experience, I've since joined Illumina, helping to work with scientists to bring the technologies that we have and being able to read DNA to help apply those to a lot of different research and applied challenges that we face. But I also like to have a bit of fun still. There's a little bit of mullet hawk, as he is described down here in me, and I still like to get out and do rock climbing and other things in my spare time. Just as well, I have two beautiful daughters who are in school at the moment, 4 and 5, and my wife is also a teacher, so we have a very strong family connection to teaching. My mother was also a teacher. I know the DNA Decoded lesson focuses around the agricultural revolution and the green revolution. And depending on how you look, there's a couple of different green revolutions. The first one being a mechanization revolution in agriculture. But the second one, which was led by this man here, Mr. Norman Borlaug, who really modernized crop breeding and crop genetics and the use of that in the 50s and 60s, and was able to really accelerate the way that we can make our crop systems much, much more productive and much more resilient to environmental stresses. And he produced several varieties of wheat and other plants that have been credited with saving as many as 1 billion people and counting by today from starvation. So, few people have had such an enormous humanitarian impact. And, seeing this and learning how he was able to help so many people through his work really gives me a lot of ownership and pride in being able to be a part of what these people do and working to help as many people as we can with the technology and resources that we have at our disposal. I really love working for Illumina and with other people at Illumina to help make those dreams a reality. But the people we work with have an absolutely enormous challenge in front of them. It turns out we have to produce more food in the next 50 years than we have in the last 10,000 years combined. And a lot of it has to do with this graph on the right here, where you can see there didn't used to be a lot of people on Earth. And just in the last few hundred years, and in part somewhat due to the agricultural revolutions that enabled us to produce a lot of food to feed them, the population has just absolutely exploded. And it's expected to get to 9 billion by the end of this decade. And people are also becoming more prosperous. And they're living longer. So there's more people eating more per person, and they're living longer as well. And so we've got a triple whammy problem that results in an absolutely huge increase in the amount of agricultural output required to properly feed all of those people. And it's still a problem today. This is not a future problem. This is a today problem, feeding the world, right? We've actually made huge progress in the last 100 years. Poverty rates have been steadily dropping. The number and proportion of hungry people in the world, which is what you can see here, has been steadily dropping. We've made wonderful sustainable development goal progress. However, COVID-19 has suddenly turned it back. And even what you'll see here is that some of that growth, even in the last 10 years, kind of started to stall out, right? We're kind of running out of productivity gains in some aspects. And so, are we at a nadir in poverty, and are we going to see an increasingly hungry world from here on out? And 800 million hungry people is enough. In fact, it's too much already, right? And we certainly don't want to be living in a world that is becoming increasingly scarce for food. And there's a lot of things that are driving this. If we think the pandemic is a problem, the twin challenges behind this of climate change and the accompanying biodiversity collapse, it's being driven by human activity on the planet. The outlook on a business as usual scale is really bleak. If we don't do anything, we're really going to ruin this planet. But it's not a lost cause. We can turn it around. There is a possibility for a healthy, productive, diverse future where we preserve our agricultural productivity and our natural environments. But in order to do this, we have to make our crops yield more per acre. We have to trade with each other so that we're producing food in the most efficient way in the most efficient places. We have to reduce the waste. We throw out an incredible amount of food, and we have to make sure that more of that food makes it into people's mouths. We have to cut the amount of animal protein and animal-derived calories. Veganism is not the solution to this, but reducing your animal caloric intake is part of the solution. We have to protect more of our environment, and we have to restore some of those environments that we've already degraded. These are already, big challenges. And genomics has a part to play. Being able to understand and potentially manipulate the genetics of the organisms that make up our environment and our ecosystems and put that food on our plate is crucial to meeting these goals and meeting these challenges. This is a nice editorial in Nature that kind of details what some of those ways are, but I'll seek to walk through them in this talk. One of the things that I wanted to make sure that I had a chance to touch on, and there's a fantastic video on this from Biocom as well, that hopefully will be sent out as well as part of the pre-reading. But really, all types of genetic modification and genetic manipulation are not genetically modified organisms. There is traditional breeding, which is guided by genetic information. So we are changing the genetics of the crops and the livestock, but we're doing it by traditional breeding. Only we're using our scientific information about the changes that we're doing by making normal crosses of plants and animals in order to do it more efficiently. And I'll talk about this, and it's been a very powerful method, and we can achieve quite significant gains even without resorting to editing or other more direct interference type approaches. One of them is mutagenesis, which is an interesting example because you're really just adding mutagens to things and changing them unpredictably. And this is actually completely unregulated. To make any change by accident is considered okay, which is a bit strange given that the other things which are much more precise and well understood are more heavily regulated. So some of the scientists working in the space have said, this doesn't necessarily make sense. This thing over here is being really, really heavily regulated, but it's actually a lot easier and simpler and more predictable than this thing over here, which is unregulated at all. Perhaps that's an argument that this should be regulated, but it's another example of how there's actually quite a double standard in the regulations provided for some agricultural biotechnologies. And this is driven largely by public sentiment rather than the actual science and risk. There are others, I'm not going to go over RNAi, but there is a significant difference between transgenics, which is what we call GMOs, and gene editing. Transgenics is about taking whole genes and whole pieces of genetic material, often even from different species, and moving them around. Turns out this is actually kind of hard to do, and it has produced some wonderful things and that we've been able to use in crop systems. But the real excitement is around gene editing because it's very, very precise, very predictable. And essentially what it enables us to do is everything we were trying to do with traditional breeding, but immediately, because traditional breeding takes a very long time. To breed a new strain of corn takes 7 to 10 years, right? To breed a new trait in an animal can take an entire lifetime. And so being able to do that almost instantaneously enables us to do a lot of things that were just previously too difficult or too expensive. But this brings up the question, should we be using these technologies? That's a good question to ask. And, I like to look back at other people in science that have, experienced significant challenges like this. And, there's no bigger example than, nuclear physics. When these guys worked out that you could split the atom, they realized how much energy that could release. And when they thought about how that energy could be used, well, they said, well, it's either a tool, it's a nuclear power plant that generates very cheap energy for a lot of people, and it's very helpful, right? It's even helping us address climate change by reducing our fossil fuel emissions. But on the same side, you can make a bomb, right? So turns out, nuclear physics isn't in and itself good or bad. It really just depends on who's using it and why, right? Every scientific tool is, we look at this quote, every man is given the key to the gates of heaven and hell. The same key opens both gates. So, agricultural biotechnology and gene editing is a tool. What matters is how it's used, not if it's used. So don't think of technology as an inherently good or bad thing. That's a moral question, right? Not a scientific one. We just have to say, well, what are the risks? Can we manage them? Do the benefits outweigh those risks? And what happens if we choose not to use this? Because there's always an opportunity cost, right? We can say we'll just be safe, we won't use it, but then maybe we could have saved 1 billion lives, like Norman Borlaug did, right? So we need to weigh these things up. I don't want to go through it in great detail, but there are a lot of positive things that the enablement of gene editing technology can bring. And we think about it in some ways. I actually just want to zoom in on this one right here. We're trying to produce more. Obviously, we need more food. But does that mean that we have to use more water? And do we use more fertilizer? And do we use more pesticide? We use more herbicide. We have to cut down more forests, all these things, right? There are negative ramifications to the intensification of agriculture. And genetics can help to address these. If we can make a plant better at taking up nitrogen, we don't need to put on as much fertilizer. If we can make that plant resistant to insects, we don't have to put on insecticide like BT cotton. And we see that, really being able to make the plants themselves superior enables us to reduce all of these other things that we've been doing in order to drive up productivity. And that's good for the environment, that's good for sustainability, it's more sustainable agricultural practices, and that helps. And that's just one of the reasons why gene editing technology has huge potential in being able to both meet that goal of feeding the planet and not destroy it in the process. But there are real risks, and I would be remiss not to call one out and discuss it. So let's look at malaria, right? So malaria, even before the pandemic happened, was its own pandemic. It was killing 500,000 people a year, thereabouts, and causing a huge amount of suffering as well of people that have malaria relapses all the time. There was a lot of reinfections, a lot of children, all in all, a really bad situation. It was absolutely amazing that we've just been able to develop the first approved vaccine for malaria, which is a huge piece of news. But there's this thing called a gene drive. It actually happens naturally in nature, but there's a way of adding a DNA piece via gene editing to something in such a way that it will be spread through the population. So you can add it to one individual, and then that individual will be more successful at producing babies than the others, and over a few generations, they'll all have it. And you can do this in a way that makes them, say, infertile, or even you could do it in a way that makes them unable to harbor the malaria parasite. So if we can engineer the mosquitoes to all not be able to carry malaria, then we solve the malaria problem, right? And maybe we can save 500,000 lives a year. Now, once you release something like this, it's just out there. You can't pull it back. So if this jumps into another species and causes a problem or something like that, you can't turn the clock back. So this is something that you have to think really, really carefully about doing, because it could be really helpful, but once that cat's out of the bag, you don't have the opportunity to put it back. And so you have to be super careful. To this date, I don't think any genetically modified or genetically engineered gene drives have ever been deployed, but they're certainly being discussed. But I do want to tell the story about traditional breeding. And let's just wind it back and say, let's not talk about genetic modification and gene editing. What have we been able to do with traditional breeding? Well, it turns out a lot, right? We really, for example, in dairy cattle, started doing it in a sort of structured way in the 40s and 50s, about around the same time that Norman Borlaug was starting to breed his wheat. And you can see that the productivity of dairy cows has gone up hugely from 4,000 liters in a year and to 18,000 liters in a year. That's a massive gain. Our dairy systems are much more productive than they used to be. That's pretty amazing. And we've done the same in plants, right? We've been able to, right at this point here, you see where Norman and everybody worked out they could really start breeding and improving these crop species very quickly. It just took off like a rocket. They've been making gains ever since, and now a field of corn, which used to produce, 20 bushels per acre, is now producing 180. That's a huge, increase in the productivity of these systems. And that's almost all through, just genetics and, agricultural practices. And the way that this works, and I'm not going to explain this in a lot of detail because it's a huge amount of horrific statistics. But rest assured, basically, we worked out through statistics how to make the desired manipulations of the animal's genetics without physically going in there and messing with them, just by saying, let's mate this cow with this cow. And we do it in a very informed way, and we're able to get lots of gain. And one of the reasons that this is really good is that you notice when we switched from just doing traditional breeding to moving into somewhere where we're using all of the genetic information in those breeding decisions, that the rate of gain starts to go up. So this is the lifetime value of an animal. And you can see it's been steadily climbing, but then it just started going at a different, even better rate of increase since genomic selection was adopted around about 2008. And this actually even came from, just go back a slide, a little bit of bragging rights, Goddard and Hayes. I didn't even know they were working on it at the time. We're actually the next desk over when I was working in Australia in that research institute. Turns out they were groundbreaking and changing the world, the way the world does agriculture, which is pretty amazing. But genomics goes beyond just breeding plants and animals, and goes into aquaculture as well. And aquaculture is interesting in that it is conducted in the natural environment, and we also fish things from the sea, and we need to make sure that our oceans are protected and that our fish stocks remain plentiful and we don't denude the ocean. And genomics can really help with this too, right? We can use genomic selection to breed farm salmon that are very productive systems. We can use genomics to make sure we're not impacting the environment around those farms. We can even manage those populations of fish living wild in the sea using genetics to make sure that we're not overfishing them, or if we're underfishing them, that we know we can take a little bit more. So there's lots and lots of ways that we can use this information. But there's a catch. In order to be able to use it, we have to understand the genomes of all of these organisms in the world. And there are hundreds of thousands of species, millions of species on the planet. And most of them we know nothing about. We've never sequenced their genomes. It's not been cost effective to do it. We did the human genome. There's hundreds of thousands of genomes left to do. So there is this ambitious project called the Earth Biogenome Project, where people have trying to cobble together the resources to go and sequence all the other life forms on Earth, so that we can use this information both to feed ourselves and to protect the environment. And a classic example of those sorts of conservation genomics projects is the success story of the Kakāpō. Kakāpō is a flightless parrot from New Zealand. It's the world's heaviest parrot. So I don't know if that's a good claim to fame, but they're pretty amazing creatures. New Zealand really had no mammals and no ground-based predators. So when predators were introduced, their numbers declined very rapidly to the point that they were actually considered extinct. But then a small population was rediscovered and moved to offshore islands. And even I had the privilege of being able to go to one of those offshore islands to help with some of these conservation projects when I was just a child. But it was very interesting. People often think, oh, this endangered species, there's only a few individuals left. Surely there's not enough genetic information for us to be able to bring it back from the brink. It's doomed. It's going to die. The inbreeding depression of marrying your cousins too many times is just going to not work, and we won't be able to save it. But what we actually found when we sequenced every single Kakāpō in existence, at the time there's only about 50, we actually found out that because the population had been so small for so long, all of those bad genes that they had actually been purged. They had been selected against very aggressively, and they weren't there anymore. And so there was actually very healthy genetics in this small population. And they were able to put in genetic breeding programs to maximize that diversity. And today, there's 4 times as many Kakāpōs, and they're still increasing. We've actually brought this all the way back from extinction, very, very close. And so genomics are really important in telling us how to save things, but also what can be saved, right? So that's very exciting. And, when it comes to humans, wildlife is not an other, it's there, we interact with it. When it's not just something we eat or an environment that we use to produce our food, we physically come into contact. And another example is even the pets that we keep. I mean, I don't know about you, but I personally have a pet gecko. It's very cute. But there's even a chance that I'm exchanging microbes with my pet gecko. And the World Health Organization has kind of recognized that, human health and animal health and environmental health are actually, all really important interconnected things, and that we have to treat them all as a unit if we want to be successful in managing any one of these things. And I think, this was really brought into sharp focus in the context of the COVID-19 pandemic, which is most likely caused by a zoonotic illness that came from wild populations of animals. And so, it's really important that we do this. And we use the same antibiotics that the animals do, right, when they need antibiotics. And overuse of antibiotics is driving a huge amount of resistance to antibiotics in our environments and in our animals, and that's transferring back to humans. And we're starting to run out of antibiotics. The bugs are starting to win because we overused these protections. And so it's very important that we're able to use genomics to monitor the levels of antimicrobial resistance and look at the transmission of pathogens from wildlife to humans, and what are the environmental things that are driving those. I mean, one of the reasons we have seen more outbreaks of zoonotic viruses is simply that humans are encroaching on the natural environment. We're moving deeper into jungles and closer to forests, and we're coming into contact with animals more, and that is increasing our risks. Also, there's just more humans to infect every single day. If you remember the graph from the beginning, the density of humans is going up very quickly. Now, I want to circle back because, interestingly, this human health and One Health idea ties back to gene editing. Yes, we can use gene editing to make animals and crops more productive. However, we can also use it for other things. Now, turns out you could use gene editing to, say, make chickens immune to influenza. Now, if the chickens can't get influenza, we can't get influenza from the chickens, and vice versa. And so the chances of another influenza outbreak go down. Maybe we prevent the next pandemic. Who knows? Is that worth using gene editing for? You be the judge, or your students will be the judge, I think is the question at hand. But we are seeing a lot of governments that initially took quite opposing views to gene editing and genetic modification, changing their rules. The UK has just opened up the use of gene editing in crops and livestock. Now that they're no longer part of the EU, which had more strict rules around that, and they think that they can make things better for the animals, make more productive food systems, and lower the use of antibiotics. And the Philippines was another example. They had a blanket restriction on GMOs, which they've just lifted. And now some of their farmers are beginning to plant GMO crops and are seeing more sustainable, larger yields. They're using less pesticides. The benefits are very clear. In particular, this one around golden rice. This was the irony. We worked with the International Rice Research Institute, which bred a particular type of gene-modified rice that would help people get their vitamin A complement, which prevents you from going blind. So, if we got a lot of people in poor areas to eat this rice, they're less likely to have blindness and vision issues as a result of malnutrition. That would be fantastic. Turns out they weren't allowed to release it in the Philippines because the government wouldn't let it, even though it was released everywhere else. And eventually, finally, they've seen the light, and now they're saying that they're allowed to do that. So we are seeing things start to go back the other way, and public support for these technologies does seem to be growing. I did want to briefly touch on environmental DNA. It does turn out that DNA is just absolutely everywhere. We're shedding DNA all the time. So is everything else. And every surface and every drop of water, and in fact, even every lungful of air has DNA in it. And so we can have a look at that DNA. When we can read it using the Illumina technologies and others, we can say what's there, even when we can't see it. And so this gives us the power to take a liter of water out of the ocean and say, there are all these species of fish and all of these species of crab and all of these species of seaweed, and they're there in these relative amounts. And that's a huge window on biodiversity. When previously we had to have people sit there and watch and see, did they observe the fish? Or things like they used to pull a whale up out of the sea and cut it up in order to understand whether or not it was there and how old it was. And now maybe we can do that without having to kill it. It enables us to do a lot of non-invasive monitoring of our ecological systems and biodiversity so that we can use those information, in preventing us from damaging the environment, knowing when we have damaged the environment, or supporting restoration projects, and we've got all sorts of fantastic community education projects around sampling of eDNA in lakes and rivers. There's wonderful community programs and even school lessons around this. It's a very accessible place for students to interact with, genomics and the environment. And it turns out that exactly the same methodology that we use to read, environmental DNA can also be used to analyze our food. It's a technique that we call metabarcoding, because we all have some conserved DNA that is the same since basically the dawn of life on Earth. Because we're all evolved from the same original progenitor organisms, billions of years ago. Some of that DNA has been so important to life that it's literally never changed. It only has small changes, and we can sequence just that barcode from any organism and identify that organism, even if we don't know that it's there. And we do that to food, it'll tell you everything that's in there. I mean, you really want to know what's in that chicken nugget? Maybe not, but we can tell you, we can tell you absolutely everything that was used to make it and how much by analyzing just its DNA. And you think that this isn't a problem? Well, they did a study when they started applying these techniques, and they looked at something like fish, for example. When you've served fish in a restaurant, you see fish in the supermarket, you have to believe what it said on the menu, what it says on the label. So how often is that true? Well, it turns out 40% of the time, and that's a lot, it's not what it says. And this is also true in herbal supplements and other food areas. There is a lot of food fraud and mislabeling or food adulteration that's going on. And a lot of it goes unchecked and unseen. And so we're now starting to see a proliferation of the use of genomic technologies in fighting food fraud and looking at authenticating food before it reaches people's plates and supermarkets. The last couple of things I just want to end on is that, it's important to me to come out and talk about these environmental projects, but I really want Illumina to walk the walk, which is why I've been really excited to see Illumina make some very significant commitments to sustainability and the environment. Recently, I think they've pledged $20 million to sustainability projects, and we've started supporting a number of those now, and I'm really delighted to be working with them to bring the power that we have and some of the resources to help some of these underfunded projects. But we've also committed to making our own operations, net zero carbon emissions. And we just made this announcement lately, and the plan that we put together for being able to achieve it has been externally verified. So I'm really hoping we can hit those goals and become a carbon neutral organization. So we can not just help the environment, but not destroy it in the process. Sounds familiar, doesn't it? And just the last slide I have is that we have been supporting projects in this space for a very long time. For the last 10 years, Illumina has been running the Agricultural Greater Good Initiative, where we seek out researchers and people who are doing super important work for the environment, but lack the resources to get their projects done, right? And to get that information out and publicly available where it can be used. And the last year's winner, well, it was technically this year's winner, was Dr. Bertram Brennick, and he's studying in bees. And, we're actually seeing at the moment a huge collapse. Part of that biodiversity collapse is the collapse of insect populations. And that comes from a lot of overuse of pesticides in agricultural systems. Starting to sound familiar? These things are all interconnected, aren't they? Well, it turns out the bees are also necessary to pollinate the crops. If we didn't have any bees around, every third spoonful of food would just vanish off your plate because we wouldn't be able to produce it. We are completely dependent on this natural system to make our crop systems work. And so we were happy to make a donation to be able to support their research projects and really look forward to the outputs of those as well. If you're ever interested, there's a whole list of past winners and all of their projects and some great videos on that on our website as well, if you're interested in the work that they do. And that is all. And I think we've got plenty of time to take questions. You're muted, Howard, if you're trying to speak.
Fantastic, Khalil. Thank you so much. We do have several questions here. Here's the first question. Do you know what the regulations are for gene editing use in the United States?
I believe it is permitted, but it's permitted under strict regulations. So I think if you need to be doing gene editing experiments and you want to, take those outside of a lab, you have to get a lot of paperwork filled out, make sure that you're covering off your risk assessments and everything else. But as far as laboratory experimentation is concerned, I think it's largely permitted. But I'm not an expert on the regulation of gene editing in the US, so please don't consider me an authoritative topic on that. I live in Canada, if anyone's wondering.
Next question here, Khalil. What type of tests are done or what is looked at in order to allow a new gene edit to be released into the environment, plant, animal, insect?
Right. Well, that would depend very much specifically on the country in question, because there are different rules in different countries. In some places it's permitted, some places it's permitted within certain restrictions. I don't think there's anywhere in the world that it's just outright permitted and you can do whatever you like. But I don't know what are those specific tests. And it's one of those things that we work with customers in, because there has been cases where there has been gene editing events and there has been off-target results. So sometimes when you do gene editing, it doesn't do exactly what it is that you thought you wanted it to do, or it accidentally did something else as well. And there have been cases where this has been discovered later on as well. So it is important that that testing is done well and that the regulations are appropriate to make sure that these gene editing experiments are properly characterized and that the risks of releasing these organisms for commercial use is well understood. But in general, for gene editing, the rules don't tend to be that much more strict in my experience than for traditional breeding, because the assumption is that gene editing makes the same edits that we would be able to fix in a population through breeding. It's just a faster way of achieving the same thing. So, something like a GMO, taking a gene from a frog and putting it in a tomato, that's not going to happen in the environment, right? But a single base change in a single gene is very common. It happens all the time. All of the plants and animals in the world, even you, are mutating all the time. You're different from your parents because you mutated somewhere along the line, everything else. This concept that the world's genetics are static and fixed and need to stay that way is wrong. It's a dynamic system that's constantly changing, right? And genetics are the same. Genetics constantly mutate and change and evolve over time. All we're doing is learning how to take the lessons from evolution and transfer them into our crops so that we can bring all of those good things together: resistance to disease, high productivity, ability to resist an insect infection, all of these, less water use, more resilience to drought, all of these things. Can we just, we found plants that have each of those things. Can we pull them all together, and do what nature could do in 100 years in just a few?
That's amazing. Here's the next question for you, Khalil. Could you elaborate more on the differences between the trigger terms of genetically modified versus genetically engineered?
I think maybe the term genetic engineering is more broad and probably encompasses both GMOs and gene editing. Maybe that is, I would need to look that up, but I think that's kind of how that works, right? Whereas gene editing is a specific methodology and genetic modification is a specific methodology, right? And just to rehash that, genetic modification is considered the moving of whole genes, rather, from one species to the next, and it is often quite random in where we put it in. In fact, the original gene editing, genetic modification experiments used something literally called biolistics. We put the DNA in a gun and shoot it. And the DNA would just go in somewhere. And then afterwards, we would have to work out, well, how many times did it go in and where did it go? Whereas gene editing sort of says, we just want to make this tiny little change here and just there. And it kind of does just that, right? So it is kind of more intentional, if you understand my meaning, than the genetic modification process, which is part of the reason why people seem generally more comfortable with gene editing than they do with genetic modification. But the difference is quite nuanced.
Thank you, Khalil. Plenty of questions coming through here. This is great. We have time for a few more here. Can you suggest some ways of conveying information about the imminent food shortage to students, and or starting the conversation about the food shortage to students who are completely unaware of it?
Yeah, I mean, a lot of us live in places that have plentiful food, right? In the US and in Canada as well. And Canada's a net food producer. I think the US is too. Literally, you produce more food than the people there consume. This is certainly not true of every country in the world. There are a lot of populous countries in the world that are net food importers, right? And without the productivity from other countries, they wouldn't be able to sustain their populations. I think it's tricky because you really just need to look at some of the demographics, right? The best resources come from people like the FAO and the UN and, the WHO, these big international organizations. They have amazing resources of databases and reports. They have all of this information. They have collated it. They have made it understandable, and it's there. If you want to ask that question, these people have made the information readily available. They're just waiting for you to ask that question. And they are there to help people and make the world a better place. These aren't the secret cabals running the world. They are really mostly just concerned people from around the world trying to come together to tackle collective global problems, and they would love to help.
So the next question here, Khalil, how do you respond to those who blame the agricultural industry progressing the effects of climate change?
Look, I think there is some, valid criticism there. The intensification of land use, mechanization of agriculture. We're burning more oil in order to produce food. We're also clearing land, and that releases greenhouse gas emissions. We know livestock emit methane, which is a very potent greenhouse gas. So, the animal agriculture and intensive agriculture does have a contributing role. That said, that contributing role is much smaller than the use of fossil fuels and energy industries. I'm not going to point the finger at someone else because agriculture still has to clean up its act. But if you think that somehow swapping everything from animal agriculture to plant agriculture is going to result in a gain, it doesn't actually work that way. Animals can be grazed on land that just can't be cropped. You can graze a sheep on a mountain, but you can't grow a crop of soybeans there, right? In fact, some level of animal involvement in agriculture is efficient, is sustainable, is part of the solution. A black and white fundamentalist approach of, let's just stop doing that one big thing and we'll be okay, is just quite a naive way to approach it. And there are sensible voices out there that are just saying, look, we are partially responsible for this, but we're also part of the solution, right? And we're just trying to feed the world, and farmers are also trying to survive. There's a lot of smallholder farmers out there that really struggle with their livelihoods. Farming is not a very profitable profession to this day and age, unless you're a mega corporation, unfortunately. And so, there are a lot of really important work being done by groups, particularly like CGIAR, to be able to make all of this technology benefit those people, the most vulnerable, the most needy, right? And not just be tools that are leveraged to maximize dollars and profit and make money. At the end of the day, hopefully we can bring everyone with us.
I agree with that. We have time for the last two questions here. You mentioned that by increasing the availability of quality food, it will lead to further growth in the global human population. Would this not contribute further to the issues of biodiversity loss, climate change, food security? How do professionals in your field discuss this issue?
Yeah, look, population growth is a challenge that needs to be addressed, but the predictions are actually, as I understand it, that population is expected to level off. And, we're actually not going to go a great deal above 9 billion, potentially, at least the way that things are going. And so we may be able to meet this goal of reaching enough productivity to sustain the population as the population itself levels off. And what's causing the birth rate to decrease, curiously enough, is the empowerment of women. Actually, we really need to empower women, particularly to be able to make their own reproductive choices, and to join the workforce and to do all sorts of things. The empowerment of women is actually one of the most strongest ways to combat climate change. When someone did a study to look at all of the different ways you could combat climate change, you could have efficient light bulbs and, green power stations and everything else. It turned out the best investment was actually a condom. And giving and helping with family planning. $1 spent with a family planning clinic is actually the most impactful climate change dollar spent, more so than planting a tree. It's amazing like that. So these things, it's amazing how they're linked to social things. So I think that problem will solve itself. And let me just take the flip side here. I mean, if the solution to the population growth problem and saving the planet is letting 1 billion people starve, I don't want to be a part of that solution.
All right, Khalil, last question here for you. Does anyone at Illumina work with any ocean-based agriculture like algae, seaweed, sea greens, or even shellfish?
Yes, yep. We have people that work in aquaculture, and they use our technologies for a number of different things. Like I said, they're using them to conserve marine environments through eDNA monitoring. We're seeing them use it to help make those aquaculture systems more productive and require less antibiotics and things. You would see the amount of antibiotics that someone like a salmon industry uses. It's obscene, and a lot of that could be reduced simply by improving the fish themselves. And so I'm deeply passionate about the ocean. I think it's absolutely important. There's a lot of amazing research going on into algal production systems and how we can have intensive cropping systems that can work in places like deserts. We have people that are specifically breeding and engineering algae in order to be able to produce them in places that simply produce nothing, right? And that's the promise of synthetic biology. All of these things of even GMOs or even gene editing and traditional breeding, we're only just starting to be able to make manipulations. What we're really aiming towards is to be able to decode all of the functional elements of biology and work out how we can rearrange them. And effectively, we can repurpose life to do any job that we need it to do. And this can solve all sorts of problems. A great example is someone that just worked out that we're going to run out of coffee. The world keeps drinking more coffee and we're going to run out of coffee. And coffee agriculture has its own environmental impacts. And so someone has now worked out how to produce coffee from a bacterium in a fermentation vat. And it tastes okay, apparently, right? And it has a much lower footprint. So I think, in a lot of ways, we're going to use biotechnology as a way of keeping some of the things that we want, but making them okay, and kind of, again, even the Impossible Burger is another great example. I mean, that's actually based on synthetic biology, where they've been able to engineer and produce literally hemoglobin, the stuff that makes blood red and makes things taste bloody, in order to make things that aren't meat taste like meat, so that we can still enjoy the taste of meat without feeling like we're destroying the planet in the process. So maybe biotech will help us have our cake and eat it too.
I love it, Khalil. So much to talk about here. I mean, you covered so much, and thank you for your time and expertise today. There's a lot of conversation here, and I hope in the next 10 minutes, we're going to share some resources to get the conversation going in your own classrooms, teachers. But thank you to Khalil for sharing your time and expertise today. This presentation will be available on our YouTube channel after this event. So thank you, Khalil. Hope you have a good day.
I will.
All right. we are going to be transitioning here in the next 10, 15 minutes, sharing some resources for you guys to be able to bring into your classroom around agrigenomics. So I'm going to share my screen. All right, first and foremost, if you haven't already, please feel free to sign up here on Illumina Care's website here that Michaela shared. Maybe we reshare here again to have access to all these wonderful resources and other events that Illumina puts on. So we're going to start off there. The second thing we wanted to share today is for those who want to have a deeper dive in your own classroom to be able to connect with a scientist with your students. Illumina has partnered with the organization called Skype a Scientist. Those who haven't heard of that, it's an organization that connects scientists to classrooms across the globe. And to do that, all you have to do is click on Get Involved and then click here as a teacher. And then you're going to go through the normal application form to request a speaker. Now, what Illumina wants to let you guys know is that you can actually request a specific Illumina scientist by going to, I'm going to show you here at the very bottom in this section where it says use this box to request a specific scientist. This is where you would type in the type of scientist you are looking for from Illumina. So I would put Illumina agrigenomics scientist. That's where you would type that in here. Make sure you fill out the rest of the form correctly. And then we will be in touch about connecting a scientist with your classroom. So another way to continue the conversation with agrigenomics in your classroom or whatever other topic in biotech life science that you would love to explore. The next resource I would like to share is an important one from DNA Decoded. So for those who don't know what DNA Decoded is, it is a partnership between Illumina and Discovery Education, where they provided a lot of resources around genomics education. The one we're going to show you today, if you go to their website, dnadecoded.org, and Michaela, maybe you could do me a favor and post that link onto the chat box so they can follow along. If you go into DNA Decoded, they have a bunch of educator resources, all free, and it's targeted for your high school classes, and they are all standards aligned and explores different genomics applications. Everything from zoonotic diseases to race and medicine, to medicines of the future, to the microbiome. And the one we're going to be exploring today is agriculture revolution, tied to some of the conversations that Khalil brought up today. But before we do that, I wanted to just let you know that all these resources are free for you to use. Each of these resources do come with lesson plans and PowerPoints that are attached to it. In this case, for Agriculture Revolution, if you click on Download Lesson Plan, it's going to take you to a PDF that has all the things you need to be able to implement this agricultural revolution lesson. Now, I'm going to dive really quick onto what this lesson is about so you can see how you can implement it in your classroom. The way this lesson is designed, it's a multi-day experience of 3 days where you're going to break up your class into groups to work on this project. Now, each lesson's around 30 to 45 minutes, depending on your class schedule. And the way this lesson is broken up is really that goal here is for you to explore gene editing. And they do that by putting in the context that students are going to be a representative of a country in the United Nations. And so the lesson is brought up in the way that day one is exploring gene editing. And to do that, they do a jigsaw article where they provide different articles around gene editing and CRISPR technologies. There are actually some videos also tied to these articles. And then day two, that is when they go in and just learn more about global hunger. And the goal of this lesson is for students to choose a country that they're going to represent. And they're going to go understand the agricultural infrastructure there, the foods that are available, and make a decision on if it's applicable for them to do gene editing to help their country. And that's where you end on day 3, where the students will convene together like the United Nations, and they'll have a debate, and they're going to share what they learned about their country, and they're going to make an ultimate vote on if they're going to support gene editing or not in their country. So that is the way this lesson is brought up. It's very applicable in your classrooms and has really nice social studies integration to it so that you can have a really good debate around gene editing and CRISPR technologies. All right. With that, I would like to turn it over to my colleague, Michaela Ortega. First, I'd like to introduce Michaela. She's new to our team and going to be helping us with the Illumina project. And she brings a wealth of knowledge from the biology world as a professor, and we welcome on our team. So Michaela, I'm going to stop sharing my screen so we can turn it over to you.
Howard. Hello, everyone. As Howard mentioned, I just recently joined the Biocom California Institute team, and I am the program manager with Biocom that is helping oversee the Illumina Genomic Discoveries project. So I'm going to share my screen here briefly. So. All right, so this was sent out. I just want to briefly run through this pretty quickly. This was sent out with your registration reminder earlier today. And if for some reason you didn't get it or the email gets lost, let me know or let us know in the chat. But I do want to highlight here are the 2 links that we referenced earlier in this presentation and in this webinar, the link to sign up for the Illumina Cares newsletter, as well as for the Biocom California Institute newsletter. And what we wanted to do is not just provide some really awesome and fantastic presentation, great information about the world of agrigenomics, but also be conscientious in providing you resources on how you can really approach this within your classrooms. So at the top here, I have gathered, and this is by no means a comprehensive list. This is just somewhere to help get you started. This is some links to various websites and programs where you can do. So I have linked here the DNA Decoded that Howard just went over, a fantastic activity. I actually do something very similar with one of my college classes in our one of the science courses that I teach. So it is a fantastic activity that I think really works well with merging together the hard science concepts with how do we even approach making decisions in the world with the knowledge that we have. So it's a great resource. We also have a few more hands-on lab activities that can also be found here, and all focused on agriculture, agrigenomics, and the like. Down here, I do want to highlight, these are just some more informational resources. So just pages with extra information, videos that you can show your students. And I do want to highlight the Genetics Literacy Project. Khalil mentioned this. A few articles that can be found on this page and part of his presentation. And as well, he talked about some other fantastic resources. So here are all the links to the various papers, news articles, or websites that Khalil mentioned or referenced during his presentation. So if there's a particular part or a bit of information that sparked your attention, here are various websites to find that. And then as well on this document, if you are interested in learning more about the Illumina Genomics Discoveries Project, DNA Decoded, connecting with Illumina, I saw there was some talks going on in the chat about getting student project proposals created and funded. Please reach out to me here at mortega@biocom.org, or you can also reach out through the Illumina Cares email as well. So please reach out to me. I'd be happy to get you connected and get your questions answered. I did see there was some talk or request in the chat as well for one of the articles that Khalil was mentioning about connecting the global warming and family planning. I did find a quick little NPR article as well as the publication. And I can add those links to this article or to this Google document as well. So please let me know if you have any questions you would like to get connected to this program, and I will pass it along or back to Howard to conclude us.
Thank you, Michaela, and thank you again, Khalil, for sharing your time and expertise today. We enjoy these conversations with teachers. We will stick around here for the next 5-10 minutes. Anybody who wants to stick around, we'll actually move you over from attendee to panelist, so we can put you on screen and we can have a conversation. For the rest of you, we will be sharing that resource once again, the Google Doc, and then once the YouTube video is up, we'll also be sharing that with you as well. Thank you again for taking time out of your busy schedules to join us. Hope you had a good time.